All right, in the next few videos, we're going to talk about the control of microbes in our environment, as well as the attempt to destroy potentially hazardous microbes that are in our environment. So what are our hazardous microbes that we are trying to control? Our targets here are things that are pathogenic, as well as things that are spoiling agents. So we're trying to prevent the spread of disease and, the spoiling of food. This is a wide array of different types of microorganisms and microbes. So we're talking about controlling bacteria, the vegetative cells, as well as the spores and endospores that we know are very resistant, other eukaryotic pathogens and spoiling agents, fungi, yeast, protozoans, worms, viruses, and also those infectious prions. So our control of microbial growth is really hitting a very wide array and diversity of different types of potential agents of infection or spoiling. The FDA and the EPA are in charge of deciding what are safe levels for human exposure. So we talk about destroying microbes in our environment, but also we know in reality that it's very difficult to eliminate all microbial life. So we have these different levels that are accepted levels, so where we're more controlling the amount of microbes that are available. So different values are given dependent on how virulent the different organisms are. So for example, like these are just a few that I've sort of picked out so you can see what kind of like units they're using. So fecal coliform, this is an indicator of if there's feces, not just human feces, it could be animal feces, potentially contaminating water sources. So safe levels of human exposure are something that's less than 1,000, and this CFU right here is colony forming units per 100 grams of water. For salmonella, it's only 3 colony forming units per only 4 grams of water. So that's a very tiny amount because salmonella are very, pathogenic, very dangerous. Viruses, and helminths, so there's like 1 egg per 4 grams of water. So the idea that we're eliminating most microbes from our environment is not necessarily true. So we have to have these standards and these levels of what is acceptable or the safe amount present, because it is very impossible in order for us to get rid of all microbes. So we have bactericidal or bacteriostatic processes, or fungicidal or sporicidal or fungistatic or microbistatic. So essentially the ending there, cidal, means to actually kill. So that is going to be describing a process or a chemical or an agent that is actually going to kill those types of microbes. Whereas if something is said to be static, that just means it's preventing its growth, right? Ideally, we're wanting to kill off these pathogens, but again, there's going to be limitations to how much we can remove from our environment, and I'll go into that later. So there are processes that lead to the death of the microbes, and then there's also ones that just will inhibit or slow the growth of whatever the microbe is that we're dealing with. Now, how does this actually work in specifically bacteria or in some other cellular microbes, right? So not viruses necessarily. So different modes of action work in different ways, but different ways in which we can kill microbes or prevent their growth is to block cell wall synthesis. So the formation of peptidoglycan is very important for microbes. So having some sort of mode of action of preventing the formation of the peptidoglycan, breaking down peptidoglycan, also disrupting the cell membrane. This is going to disrupt the membrane integrity, which will allow water to flow into and out of the cell, and eventually, cause a cell to lyse. There's these molecules called surfactant molecules. Surfactant molecules actually can get into the membrane and destabilize. So just one way in which we're acting to destroy microbial life. The other ways are to interact with the proteins or nucleic acids. So there's lots of different ways in which this can happen. You can bind to ribosomes, which can stop the translation process. You can bind to DNA, which can prevent transcription and then translation, right? You can mutate the DNA, so having some chemical that will change it. We can denature and break down the proteins. So there's all sorts of different methods. We can block active sites, right, of interacting with that protein formation and the nucleic acid formation. And again, we'll go into detail about all of these different processes in the next video. All right, so what is the result then from whatever we're doing to remove or get rid of those organisms? One is said to be sterilization. So we're just going to talk about these terms here. I'll go more into detail here in a second. So sterilization is the term that we use to say that we have destroyed all microbial life. The terms disinfection and antisepsis, that is only the destruction of most microbial life, and disinfection is used specifically when talking about reducing contamination on inanimate surfaces, so like tabletops, door handles, things like that. Antisepsis is used when describing the reduction of contamination on living surfaces. And then the term decontamination is specific to a mechanical removal of microbes. All right, so sterilization is going to remove all viable microorganisms, also including viruses. So we say microbes. And when we have sterilized something, we say that that object is now sterile, right? And this is only possible for inanimate objects. It's typically done using heat or and this is a picture here of an autoclave. Again, we'll talk more about that later. It sometimes can be done using chemical means. Disinfection is the use of some chemical agent or could be a physical process to, get rid of vegetative pathogens. It doesn't necessarily destroy bacterial spores. Again, this term is only used for inanimate objects, and we'll go through lots of different types of chemical agents and things that are used in order to disinfect. So it could be a bleach solution, it could be alcohols, it could be something that's acidic. We'll go more into detail about that later. Antisepsis is when you're applying something directly to a body surface that's going to get rid of microbes on that surface or inhibit the growth of pathogens on that surface. Before you inject somebody, right, you rub with like an alcohol swab. There's all sorts of procedures put in place when there's going to be an invasive like surgical procedure, right, to prevent infection. So just to kind of tie in, I wanted to, we've just learned this term, or we're using this term antisepsis. We have used sepsis and asepsis as terms so far this semester. So I just want to clarify all three of them. Sepsis is specifically when we have growth of microorganisms in the blood or in the tissues. Right? Aseptic technique that we've been using throughout the semester in class is all of those techniques that we're using to prevent the movement of infectious agents from one place to another. So the flaming of our loops before we're transferring bacteria from a plate, from one plate to another, for a plate to a slide or whatever, that's asepsis. So antiseptic process is like inhibiting growth on body surfaces. Sepsis is when we have growth in the blood, and then aseptic techniques are those techniques we're using in the lab to prevent the movement of bacteria around. All right, so in the next video, I'm going to go through and discuss all of these different methods that result then in one of those four things: sterilization, disinfection, antisepsis, or decontamination. And this is going to be done through different methods, either physical methods, chemical methods, or through mechanical removal. And we'll talk about each different method and what sort of results and what sort of equipment and techniques are used in order to achieve one of these four possible outcomes.